Welcome again friends. In this video tutorial we'll be talking about cytokines and their functions. More about cytokines and their cytokine receptors and their families and all these things. Okay. Now cytokines, let me first tell you, cytokines are simply chemical molecules. They are simple chemical molecules. Okay. And these molecules are helping in intercommunication, or you can say the communication between the immune cells like T lymphocytes, B lymphocytes, macrophages, and all the different cells. So it helps in the communication. So it carries, so it is acting like a chemical response from one side of immune cell to another type of immune cell. Okay, so let's begin with the different kind of cytokines. Now, first of all, you must understand what are the normal type of interaction of these chemicals, because when you're talking about chemical mediators, the first thing come to our mind is hormones. Like we all know that hormones are endocrines, sorry, they're a endocrinic activity. We all know that. Now what do you mean by endocrine, what do you mean by autocrine, paracrine, and all these things? So we need to learn all these things first. Now the chemical signal that we can talk about can be the basic of chemical reaction is simple like that. So let me take example. So let's say this is a cell. It is having a particular receptor, and there is another cell. Let's say this green cell. It is having another type of receptor, for example, and it is also. So let's say this green cell is secreting this red molecules. So it is secreting this red molecules which will come and finally attach to this blue receptor of this blue cell. Now this receptor is telling the finally different kind of downstream signaling inside the cell of this blue cell. As a result, this blue cell will do some important job. For example, say it can proliferate, it can mature, it can grow, it can divide, or sometimes it will be dead, right? So this is the basic of chemical signaling, right? So must have clear cut understanding of this chemical signaling. So for this chemical signaling we must have a chemical, what we can say, chemical donor. Here it is, the donor is this green cell, from where the chemical is originated, and obviously one receptor, right? So here it is, the receptor is this blue cell, right? So this one is the receptor in this case. So this donor is secreting those chemical molecules, receptor is receiving it using this blue receptor that you can see. So there are some chemical receptors are there, and obviously ligands are there. So these chemicals are acting as ligands, and there are receptor for all those ligands. So we are talking about always the ligand receptor interaction in immunology. Okay, so in this first case what we seeing here, in this first case we are seeing the autocrine action. In the autocrine action, it is suggesting us that we are having one cell. That cell is secreting some important mediators which is activating that cell itself. So it's a self activation. It is called autocrine. Auto means itself, so it's a autocrine activity. Now the second type is called the paracrine. In this case, one cell is secreting some important mediators which is going to act on a different cell. So this is say the cell number 1, this is say cell number 2. It is acting on a different cell. So it is secreting and acting on different cell. It is called a paracrine. So para means different from the origin, right? Now the third kind is called endocrine, which we have already talked about the hormones. They are endocrine. That means once one cell is releasing some chemical factors which are traveling throughout our circulation, then finally reach to a distant target cell. The target cell in this case of endocrine activity is placed very distant location from the source region. Okay. Now, in case of the cytokine activities, we are going to see all of this paracrines and autocrines activities going on. Now, you can see here, this is say this is the activated T cell. In all this case, we all know that this T cell usually will come and attach with the MHC2 molecule containing cell. Then it will providing in the some signal as the result of, or signal as a form of interleukins. So as they are forming signals in the form of interleukins, they are going to activate rest of the cells. So let's say this is the activated cell. It is releasing interleukin 4. This interleukin 4 on its own can activate many different cells. So here what we can see, one particular chemical is activating many different cell and releasing many different effects. And what are the effects? Effects are activation and proliferation of B cell, and this IL-4 is also helping this thymocyte for the proliferation. It is also telling the mast cell for the proliferation. So you can see this IL-4 is activating three different cells, and they are providing three different effect in all these cells, right? So it is called pleiotropic effect. Now pleiotropy mean one particular one particular cause result in three many different variety of effects. So we can see it here, due to the presence of IL-4, it is creating the effect on different cells like that. Okay, so this is one type of activity. Another type of activity we can observe is called the redundancy. Now the redundancy means we are again having activated TH cell. Now this activated TH cell is secreting IL-2, IL-4, IL-5. So in case of this pleiotropy, we know that one chemical is activating many ways, but in case of redundancy, we are going to see many chemicals are going to have a same function. This is called the redundancy, exactly opposite of the pleiotropy. Now in this case, we can see T helper cell can secrete IL-2, IL-4 and IL-5. All of them are leading the effect of the proliferation of B cells. So this is called the redundancy effect. Now the third kind of effect we are going to talk about is called the synergy. Now synergy means the synergistic effect. So in this case, again activated T helper cells can secrete IL-4 and IL-5. Both IL-4 and IL-5 together is helping the B cell for the maturation, and also they are helping this B cell to switch from class M to IgE or immunoglobulin E phase. Okay, because the activity is synergistic, because without the presence of both the activity of IL-4 and IL-5, this cell cannot achieve this process. And finally is antagonism, which is again against or opposite to synergism. Antagonism means, here let's say this IL-4 secreted by activated T helper is telling B cell for the class switch into IgE. But again, this interferon gamma is also secreted by this T helper cell along with IL-4. When the T helper cell secretes interferon gamma, it will block the class switching. When it secretes IL-4 and IL-5, it is triggering class switching. But when it secretes interferon gamma, it is giving opposite that effect and blocking the class switching. So both of these chemicals are secreted from the same cell, activated T helper, but they are acting opposite directions, right? So this is called antagonism. So you can see in all this case of chemical signaling, there are pleiotropic effects, there are redundancy, there are synergistic effects, and also antagonistic effects throughout the place. But still, all of these effects are necessary during the signaling, and how we are going to see later. Now let's say a typical example of this. So here we are having activated T helper cells, and usually activated T helper cells are secreting interferon gamma. Now once they are secreting this interferon gamma, it is activating macrophage. So this green person here is macrophage. Now it is activated by interferon gamma secreted by activated T helper cell. Now once this macrophage is getting activated, it must send some signal to T helper cell. It is sending the signal on the basis of interleukin 12. Now it is secreting interleukin 12, and this interleukin 12 will go and attach to the T helper receptors, a T helper cell, and it will tell the T helper that everything is going fine, macrophage is sending a signal, so everything is fine, nothing to worry about. And so this is a cascade which is going on and on and on. It is called the positive feedback loop. That means T helper cells are activating more and more macrophages. As they are activating more and more macrophages, all of these macrophages start secreting IL-12. So they are telling this T cell, they are activating in turn, they are call upon more and more T helper, and so on, macrophage is activating and going on. So this is a positive effect. So one result is triggering this particular cause to go again and again and again. This is the kind of positive feedback we can observe using the cytokine signaling in immune system. So now let's talk about a little bit about the cytokine receptors. Now, depend upon the, remember I've talked before that there must be two important things. One is the donor, another one is the receptor. So now we are talking about receptors. So cytokines like interleukins are acting as a ligand. Now we must have receptors for the ligand receptor binding. So for that reason, there are several variety of receptors that we are going to see. Actually, 5 different variety of receptor we are going to see. Let's begin with the first type. Now the first type of receptor we are going to talk about is the immunoglobulin superfamily receptors. We all know what immunoglobulin superfamily means. It means simply the typical kind of disulfide linked with folding of this chain we can observe in immunoglobulin family, as well as the anchored region in cell membrane. Now usually these immunoglobulin superfamily receptors are found to be present on MHC class 1 and MHC class 2 molecules, and very rare, they are found very rarely in case of this cytokine receptors, but they can be found. Here you can see only interleukin 1 MCSF, which is called monocytes colony stimulating factor, and also c-kit. Now this kind of receptors are this immunoglobulin family, which are here in this case I must write rare. Now second type we are going to talk about is a class 1 cytokine receptors, and in this class 1 cytokine receptors we can see lot of different interleukins. In fact, most of the interleukins are having the signaling via this cytokine 1 class 1 type of receptor, right? This receptor is also called hematopoietin receptor. It's also during the hematopoiesis we can find this kind of receptor coming out. Now this receptors is having two different separate domains we can see here, and in the top domain, which is the interacting domain, it is having conserved systems, and four different conserved system, two different domains. So this is a classical architecture of this class 1 cytokine receptors. You don't need to mug up all these things, so simply you try to follow the differences that we are going to talk about. So actually this type of receptors can bind with ligands like interleukin 2 to 15 you can see here, and also GM-CSF, GCSF, granulocyte monocyte colony stimulating factor, uh, granulocyte colony stimulating factor, and all these things, right? And also growth hormones like epidermal growth factors and all these things. Now the third kind is a class 2 cytokine receptor, but there is a difference between class 1 cytokine receptor and a class 2 cytokine receptor. Now class 2 cytokine receptor are called interferon receptors, but class 1 cytokine receptors are called hematopoietin. So these are difference in the naming, but we are having the difference in the structure also, because this one is having conserved domain in both the different domains. So you can see here in this the domain 1 and 2. This is also made up with two different domains 1 and 2, but they are having two conserved sequences in both this domain. But here you can see four of the sequences are present in the top domain but not in the bottom. So there are the difference here in this case. Okay, and this is as we call, this is the interferon receptor, so it will bind with interferon alpha, gamma, and beta, uh, but it also can hold on to interleukin 10. Now let's talk about the fourth type of receptor, which is the TNF or tumor necrosis factor receptor. This is completely different from the cytokine receptor class 1 and class 2, because these are having four different domains, and each domain is consisting of three different binding regions, three different conserved regions like C1, C2 and C3, right? So they are destined to bind with TNF alpha, beta, CD40 and also Fas ligands. Okay, and the final one is the chemokine receptor. Now remember, I've told many times, I've used this word chemokine. Let me write it here: chemokine and cytokine side by side. So we always kind of write these two words side by side, but we can see that there is a remarkable difference between the chemokine receptor and cytokine receptors. So don't forget this important concept. So if you're thinking about the chemokine signaling, the receptor is different. It's a transmembrane receptor. It's a 7-TM or 7 transmembrane receptor. That is the typical receptor found in the G protein signaling cascade. If you remember G protein signaling cascade, you can find it. If you don't know it, you can go back to my YouTube channel. You can find many videos on this G protein signaling pathway. You can see it. It's a typical receptor that span the cell membrane 7 times. That's why it's called 7-TM or 7 transmembrane receptor. And this receptor is can attach with a G protein, and also the G protein can be cleaved. Finally it will be cleaved into alpha and beta gamma subunit, and then there will be dissociation, then there will be further downstreaming activation of phosphorylate enzymes or kinase enzymes, right? So this kind of signaling we can see in case of insulin signaling and also glycogen breakdown and all these cases. So this is a type of signaling for chemokines, right? Like RANTES, like MCAF, NAP, and also MIP, and all these things. These are the examples of this chemokine: MIP, MCAF, NAP, and all these things, right? So these are different than those ones. So what we can see here, completely three different pores, four different types actually. Immunoglobulin type, which is very rare, having immunoglobulin fold. Second one is the cytokine receptors 1 and 2, which are having remarkable similarity but having slightly different variations. Third one is the TNF receptor. The TNF receptor we are going to see is a completely different, having four different domains instead of two, which is present in class 1 and class 2 cytokine receptors. But the fourth one is chemokine, which is completely different. 7 transmembrane receptor can bind with G proteins. So these are the variations, and all of these variations are important for different kind of signaling. Now you can see the signaling that we are going to see in case of this class 1 and class 2. cytokine receptor like interferon and hematopoietin receptors will be different than that of the chemokine receptor, right? Because the signaling and downstreaming will be different. So having these variations in our different immune cells allow us to signal in different ways, so that if one signaling is blocked due to the activity of foreign pathogen, another signaling can work, right? So that's a very important fact that we must talk about. Okay, so that's it, and I hope that's helpful. Thank you.